Hello, in this podcast we're looking at evolutionary forces in speciation, and the reason why we're looking at the forces besides natural selection and mutation is we've already looked at natural selection and mutation in a previous podcast, and we're also going to be looking at the types of speciation. First of all, we'll start with genetic drift. Genetic drift is sampling error, and I have a demo I'm going to show you right now. Sampling error is all about probabilities, and I'm going to illustrate this example of genetic drift by showing you here. We've got here 10 pennies, and with pennies, of course, we've got an equal number of heads as we have tails. Now, I've got both kinds of tails here, but I'm not going to differentiate between the new tails and the old tails. We just can pretend that they're the same. So we have equal numbers of heads and tails. So that means that we expect when I toss these 10 pennies, we expect to get 5 heads and 10 tails. Let's see what we get. Okay, so here we have the pennies, and I'm going to count them out here. I don't know if it's hard to see them all on the screen here, so alright, so here they are. We have here 1, 2, 3, 4, oh, you got exact 5. So there's actually no sampling error here at all, which is not what I was looking for. So we're going to try this again, because we'd end up, sampling error is actually where you get off from the expected. One of these fell off the table. Okay, so now let's take another look. Okay, so we have here 1, 2, 3, 4, 5, 6 heads, 4 tails. So the first time I did it, there was no sampling error. So that first time, if that had gone on to the next generation, we would have ended up with exactly the same probability of getting a heads as getting a tails, because there would have been just as many heads as tails. But the second time that I flipped it, now we have a sampling error. It's just through random chance, just through probabilities, that we didn't get exactly what we expected. We just were off by 1, where instead of getting 5 heads, we got 6 heads, and instead of getting 5 tails, we got 4 tails. Okay, excuse my handwriting here, but we're starting here with 5 heads, 5 tails. And then, in our next generation, we had a sampling error of 1. So we ended up with 6 heads and 4 tails. So that means we had deviation of 1 penny, or it was also 10%. And that amount is preserved to the next generation. But now let's say that the population expands. Let's say it grows. So instead of having a population of 10 pennies, we now have a population of 100. We have to keep the proportions the same, where we have 60% heads and 40% tails. So that means what we have, let's say we have 100 pennies, so that, or a population of 100, so that means there would be in that gene pool 60 heads and 40 tails. Now, if we have our same deviation of 1 penny, that would mean in that next generation, we would end up with, say, let's say it goes the same direction, we would have 61 heads and 39 tails. So now the error with 1 penny again, instead of being 10%, is now 1%. Here's where you get the power of genetic drift. As the population gets small, like what we have over here, A small change makes a big change in the gene pool. Genetic drift at a larger where you have a larger population, that you have a much smaller effect. So genetic drift is very powerful in small generations, and those changes get preserved when the population grows, if the population grows. And the bottom line with genetic drift is that it increases the variation or the differences between different populations. So, so if you compare populations over a wide range of areas, you'll see that the the gene frequencies in those populations would be different from each other. And we sort of see this in our own human population, in that different regions, people tend to look sort of like each other and not like elsewhere. The first example I'm going to give of genetic drift, and there are two examples, is called founder effect. This happens when a small population migrates to an uninhabited location. Because it's small, that means that the effect of genetic drift can be large, and that can completely overtake natural selection. So genetic drift can make a gene that is not all that fit very common. And one example of this is found in the Dominican Republic in the village of Las Salinas, where it's a small town, now has about 6,000 people there. And about 8 generations ago, one of its founders was a man by the name of Altagracia Carrasco, and he had several children by at least 4 different women. And he was heterozygous for a genetic disease that causes testosterone deficiency. Because it was a very small population, much smaller than it is today with 6,000 people, and because he had so many children, his genes are proportionately found much greater than would normally be found in the population of that village. So there are a lot of people with this testosterone deficiency disease. And basically what happens is that when a child is in the womb and developing, the testosterone is not enough of it is being converted into a usable form in order to produce the male characteristics. So functional testes are made, but that's it. There are no other male characteristics, and those testes do not descend, so that the baby who is XY is born looking just like a girl. Then when the child reaches puberty, enough testosterone is produced to make the testicles descend, and the male characteristics will all form, and a penis will grow. So that you end up with a girl reaching puberty and turning into a boy. When people have this disease outside of that town, it can cause serious emotional problems. But in this town, it's not a big deal because this disease is very common. If someone has this, it's not this. Same thing happened to his Uncle Sally. Because of genetic drift, you could end up with these rare genes becoming common, and you could also end up with some common genes becoming rare. As the population grows, these differences end up being preserved, and you can see that in the diagram here. Another way that you can end up with a very small population is called the bottleneck effect. And what happens is that you start with a large population, something happens to greatly decrease the size of the population. And then when the size gets very, very low, then sampling error becomes much, much stronger, and you end up with a disproportionate sample of genes. And when the population size grows again, you end up with a different population, different genes in the population. You can end up with genetic diseases which are common in one population that are rare elsewhere. We see this in Jewish populations who come from Eastern Europe, where Tay-Sachs disease is very common. And Tay-Sachs disease is a disease of the brain where a baby is born normal and dies by the age of 5. Another force in evolution is gene flow. Gene flow basically is the genes flowing from one place to another. This occurs when individuals move from one population to another. And if you look at this picture here, this is a picture of train track in Grand Central Station. And if you look at the people, you notice that there is a wide variety in the different features, more so than you would see in Manhattan, say 500 years ago. And that's because there has been a lot of gene flow into New York City. The population in New York City is very, very diverse because people live here who've come from all over the planet. The nub of this is that as time goes on here, the genetic differences between the different populations end up being reduced. So this has sort of the opposite effect that genetic drift has. Genetic drift makes populations more different. Gene flow makes them more the same. Now we come to speciation. Speciation is the creation of new species from old species. The thing that has to be done in order for a new species to appear is that a species has to be isolated long enough for differences to evolve that turn that population to a new species. And in particular, it has to be usually like a reproductive barrier has to evolve. The reason why it has to be a reproductive barrier is because one of the definitions of a species is that if two populations can reproduce, then they're still the same species. If they cannot, then they're different species. Allopatric speciation is one form of speciation, and here a geographic barrier isolates a population long enough for the populations to evolve barriers. And the example that I have here in the diagram here is we have the barrier of the Grand Canyon, where the Grand Canyon wasn't always there, so that these chipmunks on the top and these squirrels on the bottom evolved from the same species. But once the barrier of the canyon appeared, then the populations on the north rim and the south rim could no longer meet each other, could no longer mate, so that they evolved along different paths, and differences became enough that they became different species. Other barriers could be something like a founder effect, where a new island appears, and for example, say a bird flies over there in a storm, lands there, and enough of them land there that a population can exist there, and they're isolated from, say, the mainland, and a new species evolve, and that's how we end up with all the Galapagos finches. And in a situation like that, you can end up with adaptive radiation, where many species evolve from one ancestor species, and that's what you have over here. Down on the bottom, we have a common ancestor from the South African mainland, that finch, and from that finch, from the few finches that made it over from South America to the Galapagos Islands, we ended up with this huge diversity of finches. Another kind of speciation is sympatric speciation, and in here there's no geographic barrier. The two different populations are around each other. There's some other type of barrier between them. It's usually caused by an error in meiosis, which will prevent them from mating. Like if there's some sort of mutation where some individuals end up with a different number of chromosomes than the main population, they can't mate. So they can only mate with each other with those that have that error, and you end up with a whole different species. This is much more common in plants than in animals, because many plants are able to reproduce both sexually and asexually, and most animals just have sexual. But still, it does occur. One of our ancestor species most likely evolved this way because we have 46 chromosomes, and all the great apes have 48 chromosomes. And we found that 2 of the ape chromosomes merged to make a chromosome 2, and we found in our chromosome 2 sort of a remnant of the ends and middle of the actual chromosomes when they were separated in our ancestors. Now we come to our concluding questions. Number 1, what is genetic drift? Number 2, why are the founder effect and the bottleneck effect examples of genetic drift? 3, define gene flow. And 4, contrast allopatric speciation and sympatric speciation. That concludes this podcast, and I'll see you in class tomorrow.